The overall goal of this video is to describe how to develop and characterize a biological actuator using flexible PDMS cantilevers and cardiomyocytes. These actuators are then incorporated with a modified base to form a self-stabilizing swimming biorobot.
The fabrication method described here is unique, wherein the mechanical base structure is modified to form a self-stabilizing swimming biorobot, whose position can be also determined externally with magnets.
The main advantage of this technique is that the base properties can be easily altered to tune the buoyancy of the device and for self-stabilization.
The method can be applied to develop other novel devices which would require self-stabilization and flotation.
Generally, individuals new to this method will struggle because mechanically deforming the cantilevers is challenging. Due to their thin size, they can easily be torn or stuck in odd shapes. Visual demonstration of this method is critical, as steps in assembly are difficult to learn without visual aid.
To begin, place a silicon wafer on a photoresist spinner. Add a thin photoresist to the wafer and spin coat at 2000 RPM for 20 seconds to obtain a target thickness of 1.2 micrometers. Next, use wafer tweezers to transfer the coated wafer to a hot plate preheated to 120 degrees Celsius. Cover the wafer with a shallow petri dish and bake it for 10 minutes. Weigh out 6 grams of a PDMS base and combine it with 0.6 grams of a PDMS curing agent. Mix